Welcome back to the Deep Dive. Today, we're doing something a little different. Usually we're looking at the macro level, big systems, health trends, things like that.
Right.
But today, we're going to zoom in, way in. We're standing at what feels like the very interface between physics and biology.
It's good to be here. And you're right, this really is the border crossing. We're looking at how an electrical spark becomes, well, a thought, a feeling, or a memory.
That's the exact tension that just grabbed me when I was reading through the sources for this. We've talked before about the nervous system being this incredible electrical grid.
Yes.
And the action potential, that spike of voltage that travels down a nerve, it's binary. It's either on or it's off, strictly digital.
Neuron fires or it doesn't. That's the basic rule.
But our experience, human experience is so far from binary. I don't just feel happy or not happy. I can feel content or ecstatic or wistful or anxious. If the electrical signal is just a monotonous beep, where does all that nuance come from? How do you get a whole symphony of human behavior from just a single note?
That is the fundamental question of neurophysiology, really. And the answer, which we're going to unpack today, is that the electricity is just the courier.
The delivery truck.
Exactly. It's the truck rushing down the highway. But the meaningful information, the nuance, the context, the memory, that's all in the package it's delivering. And that package is chemistry.
So the signals carry the information, but the chemistry is what gives that information meaning.
I couldn't have put it better myself. Today, we're exploring the chemical vocabulary of the nervous system, neurotransmitters and neuromodulators.
And we have a pretty heavy stack of reading for this one. We're pulling from two of the absolute cornerstones of medical education, Ganong's Review of Medical Physiology and the Guyton Hall Textbook of Medical Physiology.
And they offer two very complementary perspectives. It's really useful to read them side by side.
I noticed that right away. Ganon feels like the chemist in the room. It goes incredibly deep into the molecular nuts and bolts, the specific proteins, how the chemicals are made. It's asking, what is this machine actually made of?
Whereas Guyton is more of the systems engineer. Guyton is fascinated by the wiring diagram. How do all these synapses create circuits? How do you get stability in the system? How does the brain process information without just, crashing? It's the how and the why.
So our mission today is to try and decode that functional logic. We're not just going to list a bunch of chemicals like a shopping list. We want to understand why the brain uses one chemical for, say, rapid movement and a completely different one for mood.
And why context is everything. Because as we'll see, the very same chemical can excite one cell and completely shut down its neighbor. It all comes down to interpretation.
So let's start at the most fundamental level, the hardware. The synapse. I think most of us have this mental image of neurons just touching each other like two wires being spliced together, but they don't really touch, do they?
In the human central nervous system, very rarely. There are things called electrical synapses, or gap junctions, where the cells are physically connected by these little protein tunnels. Ions can flow right through.
That sounds incredibly efficient. Why don't we just use that for everything?
It is efficient, but it has a massive flaw for a brain that needs to think and process information. It's bidirectional. If you and I are connected by a gap junction and I get excited, you get excited, but then because you got excited, the signal washes back and excites me again. It goes both ways.
It's an echo chamber. There's no forward direction.
It's perfect for tissues like the heart, where you want every single muscle cell to contract at the exact same moment. But for computation, it's a disaster. You can't process information logically if the output keeps flowing back into the input.
So the chemical synapse, that tiny gap, is what introduces the one-way street.
Precisely. Guyton really emphasizes this. He calls it the principle of one-way conduction. The signal comes down the presynaptic neuron, the sender, it hits the gap, and then it has to be chemically ferried across to the postsynaptic neuron, the receiver. It can't go backward.
It forces the information to flow towards a specific goal, whether that's a memory or a muscle movement or a decision. It imposes order on the whole system.
It creates the architecture for logic. Without that little gap, that separation, the brain would just be a chaotic storm of electrical noise.
Okay, so let's walk through that sequence. An electrical signal, an action potential, is screaming down the axon, and it hits the nerve terminal. It reaches the end of the road. What happens right at that moment?
The arrival of that voltage spike is the trigger. It literally changes the shape of specific proteins on the membrane of that nerve terminal. These are the voltage-gated calcium channels.
Right, calcium. It pops up everywhere in physiology. It feels like the universal go or do it button in the body.
It really is. In muscle contraction in the heart and right here in the brain, When those channels are triggered by the voltage change, they snap open and calcium, which is in high concentration outside the cell, rushes in.
And this is a key point from the reading. The relationship is quantitative, isn't it?
It is, and it's crucial. The amount of neurotransmitter that gets released is directly proportional to the number of calcium ions that enter the terminal.
So a small electrical spark lets in a little puff of calcium, which releases just a whisper of a chemical message.
And a massive train of electrical spikes, a high-frequency burst, floods the terminal with calcium, and that releases a huge shout of a message. It converts a digital signal, the action potential, into an analog, graded chemical signal.
So the chemical is just sitting there waiting, I assume. And those little bubbles I remember from biology class, the vesicles?
The synaptic vesicles, yes. You can think of them as these pre-packed cargo ships. Each one is loaded with about 2,000 to 10,000 molecules of a transmitter, say glutamate or acetylcholine. And they're docked right at the edge of the membrane, just waiting for the signal to launch.
And calcium is the key that unlocks the dock and pushes them off.
The mechanism is quite beautiful. The calcium ions bind to what are called release sites, which are protein structures on the inner surface of the membrane. This binding triggers the vesicle to fuse with the cell wall and essentially turn itself inside out, dumping its entire cargo into the synaptic cleft.
That's exocytosis.
Yes. And it happens in fractions of a millisecond. It is violently, incredibly fast.
So now we have thousands of these chemical molecules floating in the cleft, that tiny space between the neurons. They drift across and they hit the other side, the receiver. And this, for me, is maybe the single most important concept in this entire deep dive.
Okay, which concept is that?
The idea that the transmitter itself, the chemical, doesn't actually make the decision. The receptor on the other side does.
That is the golden rule of neurophysiology. If you remember one thing, remember that.
I can take one molecule, let's say acetylcholine. If I drop it on a skeletal muscle, that muscle contracts. But if I drop that exact same molecule on the heart slows down. The chemical didn't change at all. The listener changed.
Precisely. The receptor is the translator. It's what interprets the message. And broadly speaking, both Ganong and Guyton divide these receptors into two huge families that operate on completely different timelines.
Ionotropic and metabotropic.
A bit of a mouthful, I know, but the distinction is absolutely vital. Ionotropic receptors are the fast system. They are for when you need speed above all else.
So how do they work? What's the mechanism?
Imagine a gate or a channel that is part of the receptor protein itself. The neurotransmitter is like the key. It binds to the lock, the receptor, and the gate swings open immediately. Ions rush through. There are no middlemen. It's a direct physical opening of a channel.
It's like a doorbell. You push the button, the bell rings. It's instantaneous.
And this is what you use for things like sensory processing or motor control. If you touch a hot stove, you don't want that signal to go through a committee meeting in your brain.
No, you want it to be fast.
You want an ionotropic reflex. Open the channel, fire the neuron, move the hand. Milliseconds.
And depending on which ions are allowed through the gate, the result is different.
Exactly. We have generally two types. Cation channels are lined with negative charges on the inside, so they attract positively charged ions like sodium.
And when sodium rushes in, it brings its positive charge with it,
which excites the cell. It moves the voltage closer to the firing threshold. We call that an EPSP, an excitatory postsynaptic potential.
And the other type, anion channels.
Those allow negatively charged ions to enter, and the main one here is chloride. If you flood the inside of the cell with negative charge, you make it much harder for it to fire. You inhibit it.
So ionotropic receptors give us this rapid fire, on or off, excitation or inhibition, fast. What then is the point of the other system, the metabotropic receptors? Why would you ever want a slow receptor?
Because sometimes you don't just want to ring the doorbell. Sometimes you want to change how the entire house operates.
Okay.
Metabotropic receptors aren't channels themselves. They are coupled to another protein inside the cell called a G protein. When the transmitter binds, it doesn't open a gate directly. It nudges the G protein, and that starts a whole cascade of events inside the cell.
It sounds like a Rube Goldberg machine.
That's a perfect analogy. The receptor pokes the G protein. The G protein releases a little subunit. That subunit might drift off and open a channel somewhere else, or it might activate an enzyme. That enzyme might create what's called a second messenger, like KMP, and that second messenger activates another enzyme, and so on.
That sounds exhausting and incredibly slow.
It takes hundreds of milliseconds, sometimes seconds, even minutes for the full effect. But the trade-off is amplification and duration. One single transmitter binding to one metabotropic receptor can trigger a cascade that affects hundreds of channels. It can change the metabolic state of the cell. It can even send signals to the nucleus to trigger gene transcription, to start building new proteins.
So if ionotropic is like sending a text message, fire now,
Yeah.
then metabotropic is like installing a software update.
That is a brilliant analogy. Absolutely brilliant. Ionotropic handles the immediate data stream. Metabotropic changes the settings, the very operating system of the computer itself. And this is the basis of mood, of memory, of learning. It's how the brain physically reconfigures itself over time.
Okay, so we have the delivery system with calcium, and we have the interpreters, the receptors. Now let's look at the actual letters in this chemical alphabet. The outline groups them into categories, starting with what it calls the workhorses, the small molecule transmitters.
Right. These are the ones doing the vast majority of the heavy lifting for day-to-day communication in the brain. They are synthesized right there in the nerve terminal. It's like fast food. Made on site, used quickly, and then the components are recycled immediately.
And the undisputed king of this group seems to be glutamate.
Glutamate is the primary excitatory transmitter in the mammalian brain. Full stop. If your brain is doing something, calculating a tip, recognizing a face, planning your next sentence, glutamate is the fuel making those neurons fire.
It's the brain's gas pedal.
It accounts for something like 75% of all excitatory transmission. It is absolutely everywhere.
Now, Guénon goes into some detail on the specific receptors for glutamate. It lists AMPA, kainate, and NMDA. And it really seems like the NMDA receptor is a special case. It's not just a standard doorbell, is it?
The NMDA receptor is a molecular masterpiece. It's absolutely critical for memory and for what we call neuroplasticity, but it's tricky. It has what's known as a magnesium block.
What does that mean exactly, a magnesium block?
Okay, so imagine the receptor is a tube, a channel. At the resting state, when the neuron is just sitting there quietly, a magnesium ion physically sits right inside that tube, plugging it up like a cork in a bottle.
Okay.
So even if glutamate comes along and binds to the outside, which is like turning the key in the lock, the door can't open because the cork is still stuck in there. Nothing can flow through.
So the key works, but the door is like boarded up from the inside.
Exactly. Now, to knock that magnesium plug out of the way, the cell membrane itself has to be depolarized. It has to be electrically excited first. And usually this happens because the other glutamate receptors, like AMPA, have already fired and let some sodium in.
Okay, so wait, for the NMDA receptor to actually work, you need two things to happen at basically the same time. You need glutamate, the chemical signal, to be present, and you need the membrane to already be electrically active.
Yes. It is a coincidence detector. It only opens if event A, the cell is already active, and event B, the new signal from glutamate arrives, happen together.
That sounds like the literal definition of associative learning. You know, the old saying, neurons that fire together wire together.
That is precisely what it is. This receptor is the molecular hardware of learning. When the NMDA receptor finally does open, it lets in a flood of calcium. And as we said, calcium is the do-it signal. The calcium triggers long-term changes that make that specific synapse stronger, more sensitive, essentially permanently. It tells the cell, this connection is important. Pay attention to it. Keep it.
That is incredible. A single protein that acts as a logic gate for forming memories. But there's a dark side to this, isn't there? The clinical box in Guénon mentions excitotoxins.
Yes. Glutamate is a powerful tool, but it's a double-edged sword. Because it lets in so much calcium, and calcium turns on all this cellular machinery. If you have too much glutamate, you get way too much calcium.
And the machinery just goes into overdrive.
It starts activating enzymes that literally digest the cell from the inside out. This is the central tragedy of a stroke. When a blood clot cuts off oxygen flow to a part of the brain, the pumps that normally vacuum up excess glutamate from the synapse fail.
So the glutamate just builds up and hangs around.
It floods the synapse. The neurons are relentlessly overstimulated. They open their NMDA channels, massive toxic amounts of calcium rush in, and the neurons literally excite themselves to death.
Wow. So a huge part of the damage in a stroke isn't just the lack of oxygen itself, it's this secondary chemical storm.
That's right. Which is why there's a huge amount of research into NMDA antagonist drugs that can block that receptor to try and stop that destructive cascade during an ischemic event.
The glutamate is the gas pedal, and it's a very powerful one. What about the brake?
The main brake in the central nervous system is GABA. That's gamma-aminobutyric acid.
And I'm guessing if glutamate opens channels for positive ions to excite the cell, GABA must do the opposite.
It does. GABA primarily binds to ionotropic receptors that are selective for chloride ions. And chloride, chelinus, carries a negative charge.
Now, can you help me with the physics here? The text brings up the Nernst potential. Why exactly does opening a chloride channel stop the cell from firing?
It's a really clever bit of electrical clamping. So a resting neuron sits at a membrane potential of about, say, minus 65 millivolts. The equilibrium potential for chloride, the voltage where it's perfectly happy with no net movement in or out, is about minus 70 millivolts.
So they're already pretty close to each other.
Very close. But remember, to fire an action potential, the cell's voltage needs to go up from negative 65 milliV to a threshold of about negative 45 milliV. When GABA opens all the chloride channels, it essentially locks or clamps the membrane voltage down at negative 70 milliV. So even if glutamate is trying to push the voltage up, the open chloride channels create this massive leak for negative charge, which keeps dragging the voltage back down.
It anchors the cell. It basically says, you are not moving from this spot.
Exactly. It creates a powerful inhibitory wall.
This brings us to some very common drugs. Benzodiazepines, for example. Valium, Xanax, they work on the GABA system, right?
They do, but they're fascinating because they aren't direct agonists. They don't open the channel themselves. If you took a drug that just ripped open all your GABA channels, your brain would shut off instantly. You'd go into a coma.
Right, not ideal.
Benzodiazepines are what we call neuromodulators. They bind to the GABA receptor, but at a different spot than GABA itself. And by binding there, they change the shape of the receptor just enough so that when GABA does come along, it binds more effectively.
So they don't open the door, they just kind of grease the hinges.
That is the perfect metaphor. They make the brain's natural braking system more efficient. So you get a calming effect, an anti-anxiety effect, without necessarily turning off the lights completely, unless, of course, you take far too much.
Okay, we've got the accelerator with glutamate and the brake with GABA. There's one more workhorse I want to touch on before we move on, acetylcholine.
The utility player. Acetylcholine is just everywhere. It's at the neuromuscular junction, so every single time you move a muscle, that's acetylcholine. It's a huge part of the autonomic nervous system, slowing your heart, digesting your food, and it's all over the brain, involved in learning, memory, and alertness.
What really stood out to me about acetylcholine in the reading was the cleanup process. With the other transmitters, the books talk about reuptake, basically vacuuming the chemical back up into the presynaptic terminal, but acetylcholine gets destroyed.
Yes, and this is a key functional difference. Especially at the neuromuscular junction, you need absolute speed and precision. If I tell my finger to tap, I need that signal to start and stop almost instantly. I can't have the chemical lingering around in the cleft, causing the muscle to keep twitching.
So how does the body handle that for such a quick off switch?
There's an enzyme called acetylcholinesterase sitting right there in the synaptic cleft. It is one of the fastest, most efficient enzymes known to biology. As soon as a molecule of acetylcholine binds to the receptor and does its job, this enzyme grabs it and chops it in half into acetate and choline. Signal terminated.
Instantly. This ensures the muscle is ready for the very next signal within just a few milliseconds.
Yeah.
And this is where things like nerve gas come in, right? Chemical weapons like sarin.
Unfortunately, yes. Many of those horrific nerve agents and also many pesticides work by being irreversible inhibitors of acetylcholinesterase. They block the cleaner.
So the acetylcholine just stays there.
It just builds up and up and up. It keeps stimulating the receptors over and over and over. The muscles go into what's called tetany, uncontrollable rigid contraction. The autonomic system goes haywire. The diaphragm, your breathing muscle, locks up. It is a horrific demonstration of just how critical that off switch really is.
It really highlights that the off signal is every bit as important as the on signal.
Absolutely. Physiology is always, always about balance. If you break the balance, you break the organism.
Let's shift gears a bit. We've been talking mostly about this point-to-point communication. You know, neuron A tells neuron B to fire.
Yeah.
But the next section in the outline moves us to the context setters, the monominds.
Yes. This is where we leave the world of specific wiring and we enter the world of more like volume control or even atmospheric conditions.
The text describes these as diffusely connected systems. What does that actually look like in the brain?
It's a fascinating architecture. Imagine a very small cluster of neurons, maybe just a few thousand of them, located deep in the brainstem. But their axons, their output wires, travel up and branch and branch and branch again, ultimately covering vast areas of the brain. A single one of these neurons might make contact with hundreds of thousands of other neurons all across the cortex.
So it's not a targeted message, it's like a sprinkler system.
That's a perfect analogy. They aren't sending a specific, discrete message like move your left toe. They are spraying a chemical that changes the entire atmosphere of the cortex. They set the weather for the day.
Let's look at the first one of these weather makers, norepinephrine.
This system originates in a tiny spot in the pons called the locus coeruleus, which means the blue spot. And from there, it projects just about everywhere.
And what's its function? What weather does it create?
Alertness, arousal, attention. When you hear a loud, unexpected noise and suddenly you are hyper-focused and your heart is pounding, that is your locus coeruleus flooding your brain with norepinephrine. It increases the signal-to-noise ratio, making your neurons more ready to respond to relevant stimuli. It wakes up the whole cortex.
And structurally, this is very similar to adrenaline, right?
Very closely related. Adrenaline is epinephrine. Norepinephrine is also called noradrenaline. In fact, there's a really interesting quirk in how it's made. Remember, we said these small molecules are made right in the terminal?
Yeah.
Well, usually the whole synthesis process happens in the cytosol, the soup of the cell, and then the finished product gets packed into vesicles. But norepinephrine is different. The very last step of its synthesis actually happens inside the vesicle.
Wait, really? How does that work?
The cell makes dopamine first, then it pumps that dopamine into the vesicle, and inside that little bubble, there's an enzyme waiting that converts the dopamine into norepinephrine. It's the only transmitter made this way.
That implies that dopamine is the precursor molecule.
It is. It's an assembly line. An amino acid, tyrosine, becomes dopa. Dopa becomes dopamine. If you're a dopamine neuron, you stop there. If you're a norepinephrine neuron, you just add one more enzymatic step inside the vesicle.
That is just beautifully efficient. Let's talk about dopamine then. Most people know it as the reward chemical.
That's the pop culture label, and it's not wrong, but it's incomplete. Dopamine is really about salience and control. Salience meaning what's important, what to pay attention to.
Okay, explain the control part first.
Well, look at the anatomy of the system. One of the major dopamine highways is called the nigrostriatal pathway. It connects a part of the midbrain called the substantia nigra to a motor area called the striatum. This entire circuit is dedicated to regulating motor movement.
And when that pathway dies?
We get Parkinson's disease. In Parkinson's, the patients aren't just sad or unmotivated, they are physically frozen. They have rigidity, tremors, and inability to initiate movement. It shows us that dopamine acts as the grease for the motor system. It allows the go command from the cortex to be executed smoothly.
But then you have this completely different pathway, the mesocortical one.
This is the one that projects to the limbic system, the emotional brain, and to the frontal lobes. This is where we get motivation, reward prediction, error, and addiction.
And this is where schizophrenia seems to enter the picture.
Yes. The dopamine hypothesis of schizophrenia in its simplest form suggests that there is too much activity in this system, specifically at the D2 family of receptors.
So if Parkinson's is, in a way, too little dopamine in one circuit, schizophrenia is, in part, too much dopamine in another.
Or, maybe more accurately, too much signal in the wrong place at the wrong time. If dopamine's job is to tell your brain, hey, this is important, pay attention to this, and you have way too much of it, you might start assigning immense meaning and significance to random things. The pattern of cracks on the sidewalk, a static noise on the radio.
That's a fascinating way to conceptualize hallucinations, as a disorder of assigning significance to noise.
That's one way to think about it, yes. A dysregulation of salience.
And finally, the third of these big monoamine weather makers, serotonin.
The stabilizer. This system originates in the raphe nuclei, which is a thin seam of neurons running down the midline of the brainstem.
Raphe means seam, right?
It does. And from this seam, these neurons project to the entire brain and also down the spinal cord. Serotonin has a hand in regulating mood, sleep-wake cycles, and even pain perception at the level of the spinal cord.
And this, of course, is the target of the most common class of antidepressants, the SSRIs. Selective serotonin reuptake inhibitors.
Right. And now that we understand the basic machinery of the synapse, we can see exactly what they do. They don't make more serotonin. They block the transporter protein, the little vacuum cleaner called SERT, that's responsible for sucking the serotonin back up out of the cleft.
So the serotonin that's already been released just gets to hang out in the synaptic cleft for longer.
It lingers. It gets to bump up against the postsynaptic receptors a few more times before it's eventually cleared away. It effectively amplifies the natural signal that's already there.
It's amazing when you step back and think that so much of modern Psychiatry treating depression, anxiety, psychosis comes down to tweaking the volume knobs on these three diffuse chemical systems.
We aren't really rebuilding the brain with these drugs. We're just modulating the modulators.
Okay, so Section 4 in our outline is titled The Rule Breakers. We've talked about the small molecule workhorses like glutamate and GABA and the monoamine modulators. What makes neuropeptides so different?
It all comes down to logistics. Everything we've discussed so far, the small molecules, the monoamines, they're all synthesized right there in the nerve terminal. The factory is located right next to the loading dock.
Very convenient.
Neuropeptides are completely different. They're small proteins, and you can't make proteins in the axon terminal. You need the whole cellular machinery, the nucleus with the DNA, the ribosomes, the Golgi apparatus.
And all of that is way back in the cell body, the soma.
Exactly. So neuropeptides have to be manufactured in the soma, packaged into special large vesicles, and then physically transported all the way down the axon to the terminal.
And axons can be really long.
In your sciatic nerve running down your leg, an axon can be 1 meter long. That is an enormous journey for a microscopic vesicle. The process is called axonal streaming, and it can take days.
So this immediately implies that neuropeptides are metabolically expensive. You can't just make them and waste them?
Correct. And another key difference. Unlike the small molecule vesicles, these large vesicles are not recycled. Once a neuropeptide is released and does its job, the vesicle is gone. The peptide is broken down by enzymes. It's a one-time use item.
So why on earth does the brain use them? What is the advantage that's worth all that effort?
Potency and permanence. Neuropeptides, things like substance P, which signals pain, or endorphins, which are the body's natural pain relief, are effective at incredibly low concentrations. We're talking maybe 1,000 times more potent than something like acetylcholine.
A small package with a very big punch.
And their effects last. They almost always act through those slow metabotropic G protein-coupled receptors. They trigger long-term changes in the metabolic machinery of the target cell. They can alter gene expression. They aren't sending a text message, they're sending a legal contract that changes the rules.
Now, this led to a concept in the reading that totally blew my mind, the idea of co-transmission. I always learned in school that a neuron is a dopamine neuron or a glutamate neuron, one cell, one chemical signal.
That's the old dogma, often called Dale's principle. And it turns out it's not quite true. We now know that many neurons contain both a classical small molecule transmitter, like glutamate, and a neuropeptide stored in separate vesicles.
Or bilingual.
Yes. But here's the incredibly cool part. They speak different languages depending on how loud they're yelling.
Okay, explain that.
It's all about frequency dependence. If the neuron is firing at a low frequency, just a slow, steady tap-tap, the calcium levels that come into the terminal stay relatively low and localized right near the membrane. This is enough to trigger the release of the small vesicles containing the fast transmitter, like glutamate.
That's the whisper.
That's the whisper. But if that same neuron gets really excited and fires a high-frequency burst spurt, the calcium levels spike much higher and flood the entire terminal. This high global calcium concentration is required to mobilize and release the large, dense core vesicles containing the neuropeptides.
So the chemical message the neuron sends literally changes based on the urgency of the electrical signal.
Exactly. A low urgency signal just says, here's the basic data. A high urgency signal says, here's the data, and here's a modulator to change the state of the entire local system.
That adds a whole new dimension to neural coding. It's not just where the signal goes, but the very rhythm of the signal determines the chemical cocktail that gets released.
It's how the brain gets immense complexity from simple building blocks.
And then we have the ultimate rule breaker, the gas, nitric oxide, or NO.
This one drove physiologists crazy for a long time because it doesn't behave like anything else. First, it isn't stored in vesicles.
How could you store a gas in a little lipid bubble?
Not easily. It would just diffuse right out. So nitric oxide is synthesized on demand, instantly. When the postsynaptic neuron is stimulated, especially by that calcium influx through NMDA receptors, an enzyme called NO synthase switches on, makes the gas, and the gas just diffuses right through the cell membrane.
Like a ghost walking through a wall.
Exactly. It doesn't need a receptor on the surface. It drifts into neighboring cells, including the presynaptic terminal it just came from, and affects their enzymes directly. But the most critical role for NO is that it can travel backward.
This is retrograde transmission.
Yes. The usual flow of information is presynaptic to postsynaptic, but here, the postsynaptic cell, the receiver, makes this gas, and it drifts back to the presynaptic cell, the sender.
What's the message it's sending back?
In essence, the message is, I heard you. It's a confirmation receipt. It tells the sender, that signal you sent was strong and effective. It caused a big calcium spike in me. So strengthen this connection. Make it easier to send this signal next time. This feedback loop is a vital component of long-term potentiating, which is the physical basis of memory.
So without that little puff of gas drifting backward, the sender wouldn't get the feedback it needs to reinforce the synapse.
Exactly. It's a critical part of closing the learning loop at a molecular level.
This brings us perfectly to Section 5, the mathematics of processing. Because we've now talked about all these chemicals, excitatory ones, inhibitory ones, slow modulators, even gases. But a single neuron in the brain is on the receiving end of thousands of these signals all at once.
A single motor neuron in the spinal cord, for example, might have 10,000 synaptic buttons on its surface.
10,000.
Wow.
And some of them are screaming fire with glutamate, and others are screaming stop with GABA. How on earth does that one neuron decide what to do?
It acts as a tiny, incredibly sophisticated analog computer. It performs a process that physiologists call integration. It literally just sums up all the incoming voltages.
So an excitatory signal, an EPSP, adds a small positive number to the voltage, and an inhibitory signal, an IPSP, adds a small negative number.
And the neuron just keeps a running tally. If the total membrane potential rises from its resting state of negative 65 milliV all the way up to the threshold, which is usually around negative 45 milliV, then the gun goes off. And action potential is generated and fired down the axon. If the sum doesn't reach negative 45 milliV, then silence. Nothing happens.
Now, Guyton breaks this addition process down into two types, spatial summation and temporal summation. Let's distinguish those.
Spatial summation is like taking a vote across a physical space. You have inputs arriving at many different places on the neuron all at the same time. If one excitatory terminal firing isn't enough to reach threshold, maybe 20 of them firing simultaneously at different spots will be, their individual small voltage changes add up, 1 + 1 + 1. The sum might be enough to reach the threshold.
Strength in numbers across space.
Temporal summation is different. It's about frequency over time. Imagine one single excitatory terminal firing repeatedly, incredibly fast. Bam, bam, bam. The first pulse causes a small rise in voltage, and before that voltage has a chance to fade back to rest, the second pulse arrives and adds on top of it, and then the third adds on top of that. They piggyback on each other.
Like trying to kick a door down. One kick might not do it, but 10 rapid-fire kicks in 2 seconds might just break the lock.
That's a perfect description. The neuron integrates both space, how many inputs are active, and time, how fast are they firing.
But there's a catch, isn't there? Not all of these votes are created equal. The outline calls this the geography of influence.
This is a key insight from Guyton. The neuron has a physical shape. You have the dendrites, the tree-like branches where most inputs arrive. You have the soma, the cell body, and then you have the axon, the output wire. The final go or no-go decision to fire an action potential is made at a very specific point, the axon hillock, the very beginning of the axon.
The initial segment.
Right. Now, electricity flowing through a cell is leaky. A current degrades over distance. So you have an excitatory input way out on the very tip of a long dendrite. That little voltage change has to travel all the way to the axon hillock. It loses a lot of its strength along the way.
So a vote from way out in the suburbs of the neuron counts for less than a vote from downtown, right next to the decision-making hub.
Precisely. Inputs that are physically closer to the soma or the axon hillock have a much more powerful influence on the outcome. And here is the strategic brilliance of brain design. Where do you think most of the inhibitory GABAergic synapses are located?
Well, if I were an engineer trying to control the system, I'd put the main stop button right next to the trigger.
And that is exactly where they are. Inhibitory synapses are often clustered right on the soma and especially at the axon hillock itself.
So you could have 1,000 dendrites out there screaming, fire, 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 but one or two well-placed inhibitory signals right at the neck can veto the entire thing?
It acts as the final gatekeeper. It can shunt the current and clamp the voltage right at the choke point where the decision is executed. This allows for incredibly precise control, like the ability to stop a planned movement halfway through because you suddenly see an obstacle.
That is some seriously elegant design logic. Now, we have to talk about stability, which is section 6. We have all these excitatory loops. Neuron A excites neuron B, which excites neuron C. What happens if neuron C has a connection that comes back and excites neuron A again.
That is what's called a reverberatory or oscillating circuit. It's a positive feedback loop.
Like a microphone getting too close to a speaker and you get that horrible screeching sound.
That's the exact same principle. In the brain, controlled reverberation is actually very useful. It's thought to be the basis for rhythmic processes like breathing. It's part of how we maintain wakefulness. But if it becomes uncontrolled,
That's a seizure.
that is the very definition of an epileptic seizure. Uncontrolled, synchronous, high-frequency firing that starts recruiting all its neighbors and sweeps through the brain like a forest fire.
So the obvious question is, why aren't we all having seizures all the time? What keeps this incredibly excitable system stable?
There are two main protective mechanisms. First, as we just discussed, there are powerful inhibitory circuits. We have feedback loops that are negative. If I start firing too much, I also excite a little inhibitory inner neuron, which then comes back and tells me to shut up. It's built-in self-regulation.
And what's the second mechanism?
Synaptic fatigue. This is the ultimate safety valve of the nervous system.
Is this related to what we said earlier about the vesicles being finite, running out of ammo?
Yes, that's a huge part of it. A neuron only has a finite supply of neurotransmitter in its ready-to-release pool. If a circuit fires excessively, hundreds of times per second, as in a seizure, the neurons simply cannot synthesize, package, and move vesicles to the membrane fast enough to keep up. The vesicles are depleted. The receptors on the other side also start to desensitize. The signal gets weaker and weaker until it finally just stops.
So a seizure will often stop on its own because the brain literally runs out of chemical gas to sustain the firestorm.
Exactly. Fatigue is a crucial protective mechanism that prevents the brain from burning itself out. It's a failsafe.
There's also a fascinating note in Guyton about the overall chemical environment, specifically pH.
Oh, this is a classic medical school pearl, and it's clinically so important. The synapse is incredibly sensitive to the acid-base balance of the blood and cerebrospinal fluid.
Alkalosis versus acidosis.
Right. Alkalosis, a high pH, meaning less acidic, dramatically increases neuronal excitability. If the body's pH rises from its normal 7.4 up to 7.8 or 8.0, neurons can start firing spontaneously without any input.
And Guyton suggests a little clinical thought experiment for this.
The hyperventilation test. If a patient voluntarily breathes really, really fast and deep, they blow off a lot of carbon dioxide. CO2 dissolves in water to form carbonic acid. So if you lose a lot of acid, your blood becomes more alkaline.
So by hyperventilating, you are actively raising your brain's pH.
And in a person who is susceptible to epilepsy, that change in excitability can be enough to precipitate a seizure right there in the doctor's office. It lowers the firing threshold for the whole brain.
And the reverse is true for acidosis.
Yes. Acidosis, a low pH, severely depresses neuronal activity. If the pH drops from 7.4 down to 7.0, the system begins to shut down. This is what we see in things like severe diabetic ketoacidosis. The blood becomes dangerously acidic, the neurons stop firing properly, and the patient can slip into a coma.
It's terrifying, really, how narrow that physiological window is. A little shift up, you risk seizures. A little shift down, you risk a coma.
We are walking a very fine tightrope of homeostasis every single minute of our lives.
We're coming toward the end here, but we can't possibly leave this topic without talking about plasticity. We've described all this hardware as if it's fixed in place. But maybe the most important thing about the brain is that it changes.
This is the so what of this entire conversation. This is why it matters to everyone, not just medical students. The synapse is not a soldered joint in a circuit board. It is a living, breathing, adapting interface.
How does it adapt? What are the mechanisms?
It's all about regulation. We touched on this with the receptors. It's called upregulation and downregulation. If a synapse is underused, if it's not getting much signal, the postsynaptic cell will often build more receptors and insert them into the membrane. It's becoming more sensitive. It's trying to hear the whisper.
And if it's overused?
Yeah.
Constantly being blasted with a signal.
If a synapse is constantly flooded with transmitter, the cell protects itself by pulling receptors out of the membrane and digesting them. It downregulates. It's turning down the volume to avoid damage.
This perfectly explains the concept of drug tolerance, doesn't it?
It's the primary mechanism. If you take a drug that floods the reward system with, say, dopamine, the brain responds by saying, whoa, this is way too loud, and it removes dopamine receptors. Now, to feel normal, you have fewer receptors. So when you stop the drug, normal life feels dull and gray. And to get that high again, you need to take even more of the drug to shout at the few receptors you have left.
You have physically changed the chemical architecture of your brain.
But this is also how we learn and get better at things. Long-term potentiation, using those NMDA receptors, using nitric oxide strengthens the connections that are useful. We physically build new receptors and new release sites at the synapses we use the most. We are constantly tuning our own hardware.
So to try and bring this all together,
We started with a simple binary electrical spike. We saw how it was transduced into a puff of calcium entering the cell. That calcium then released a specific chemical, a word, into the synaptic gap.
and that word was interpreted by a receptor on the other side, either a fast doorbell, the inotropic kind, or a slow system-wide renovation, the metabotropic kind.
That one signal was then integrated mathematically with thousands of other signals. It was modulated by the weather systems of the monoamines, and it was subject to the final veto power of inhibition right at the axon.
And finally, the history of all that activity physically changed the hardware of that synapse, making it either easier or harder to fire the next time.
That's the entire journey, from a millisecond electrical event to long-term structural change.
It really makes you realize that thinking isn't just electricity. It's this incredibly rich, messy, beautiful, and tightly regulated chemical soup.
And it means that what you do and what you pay attention to truly matters, because the things you do repeatedly are what your brain physically and structurally reinforces. You are, in a very real sense, the architect of your own mind.
That is a very powerful place to leave it. For those of you listening, I really encourage you to look at the source material for this one, specifically the drug tables in Ganong and the diagrams of summation in Guide to Kind, and check the show notes for links. Seeing the geometry of the synapse really helps all of this stick.
Absolutely. The diagrams are essential for understanding the spatial relationships.
Thanks for diving in with us today. We'll see you on the next one.
Goodbye.